Hi everyone, and welcome to our video on the effect of pH on enzyme activity. So this is the fifth one in our little enzyme series, and we're going to be having a look at the spec reference 2.1.4d, which is the effect of pH on enzyme activity, surprisingly. Let's start with the most basic recap that I hope is almost insulting to you with its ease, which is just what different pHs relate to in terms of acid, alkaline or neutral solutions. So pH 0 to 6 is our acid, 7 is our neutral, 8 to 14 is alkaline. Please make sure you know that because it's kind of a year 7 concept, so I really hope I am just teaching you to suck eggs at this point. The next thing we need to understand is moving a little bit further on, which is what happens to acids. Now, when we're looking at an acid in a solution, then what's going to happen is it's going to dissociate to form two oppositely charged parts. So we will form protons, which are hydrogen ions, and we can see our little hydrogen ion right here, H+, and it will then form a negative ion, which is the remainder of its actual compound. So if we've got hydrochloric acid, this is our top one here, then HCl, the hydrogen ion, becomes the proton here, and the chloride ion is the other part, the negative ion. If we look at sulfuric acid, H2SO4, we then dissociate to form our hydrogen ion, and of course we are going to have two of those because we've got H2, and then we are going to have our sulfate ion, SO4-. So always forming our proton, H+, and we're going to get the negative ion the rest of the molecule. When we're considering pH and its effect, we need to also understand basically how we can resist these changes in pH. Because where we're talking about living organisms, they don't want to try to deal with a range of pHs because it just wouldn't work. It's going to have a negative impact on enzymes if the pH fluctuates dramatically. Therefore, they need some kind of method to prevent this. And the method they use is in something called a buffer. Now, buffers are just substances that resist changes in pH. And one that you are going to look at through your A-level biology course is hemoglobin. Now, hemoglobin is that part that we find within our red blood cells, our erythrocytes, and this is able to act as a buffer. You're going to find out how a bit later on in the course. But because we've got this hemoglobin that can act as a buffer as part of the blood itself, that means that we can have this resistance to pH changes in the blood. And that's because a buffer can either donate hydrogen ions, so basically release those hydrogen ions into solution, or they can accept hydrogen ions, so remove them from the solution. So depending on the scenario is going to tell us what happens to keep it within a narrow range of pH. This ties really nicely into some of those practical considerations. Now, if we're doing any kind of enzyme reaction that isn't looking for a change in pH, then what we would need to do is make sure we control the pH. And the way that we do this is using a buffer to keep that pH constant. And it's worth just bearing that in mind as one of those techniques that we can use as a control variable. Read whatever experiment it is. If it could involve a change in pH, then we need to control that. How do we control it? We use a buffer to keep the pH constant. If we now consider what happens if the pH wasn't controlled. So let's start off with the concentration of our hydrogen ions increasing. So in the scenario where the concentration of hydrogen ions increases, what we're going to see is hydrogen bonds and ionic interactions in the tertiary structure of our protein, that three-dimensional shape, will become disrupted. Now, because we've disrupted the hydrogen bonds and the ionic interactions there, what we're going to see is a change in the shape of the molecule. So because we're changing the shape of a molecule and we're talking about enzymes, that means the active site has changed its shape. Because of that, it's no longer complementary to the substrate, and therefore the rate is going to decrease. The reason it's going to have a second impact is because if we're talking hydrogen ions, remember, they have got a positive charge on them. Within our actual enzyme, we are going to have a range of R groups, those little side chains on the amino acids that make it up, and some of these could have a negative charge. Now, if we've got an R group with a negative charge, what's going to happen is those hydrogen ions, which are positive, will be attracted to those R groups in the active site. Now, if you imagine what we've now done is attach hydrogen ions onto those R groups, we've also created a little bit of a barrier, if you like, for the substrate. So we've just put a little bit of a barrier in the way, making it harder for the substrate to bind, because the hydrogen ions are now binding with the negative charges of the R groups. Because of all these impacts, what we find is the enzymes only work in a narrow pH range. So when you actually look at your typical little graph, you're going to get this quite standard little bell-shaped curve. Now, what we have, obviously, we still have our optimum, which is going to be the pH at which we have the maximum rate of activity of our enzyme. But if we shift just a small amount in either direction from that optimum, either to acidic or to alkaline, then we see reductions in the rate of reaction because of that disruption to the active site. If we can restore normal pH, those bonds can reform, the active site can return to its original shape, and we can restore that activity of the enzyme. However, if we've got an extreme shift in pH, so we're going a little bit further than just a teeny variation either side, then what we're going to find, those changes to the active site are likely to be permanent changes. And at that point, we say the enzyme is denatured. And that happens if it's either becoming too acidic in the environment or too alkaline. The same effect. Enzyme denatures, active site has changed shape permanently, substrate no longer fits. Final point to remember about our enzymes is that they do not all have the same optimum pH. Our different enzymes are designed to work in different regions. So intracellular enzymes, the ones that are designed to work within the cell, these are going to have an optimum pH of around about 7. Extracellular enzymes, the ones that we're going to be secreting into a different environment, these will have varying optimum pHs depending on where they're being secreted to. So a good example to remember here is pepsin, which is an enzyme that's designed to work within the stomach. Now, the stomach obviously has that hydrochloric acid within, and therefore it has got an acidic pH. It's around about pH 2 in there. So because pepsin is designed to work within the stomach, that means it has an optimum pH of around 2 as well. And we can see that on the little graph at the bottom, two different enzymes. We've got pepsin on the left, trypsin on the right, and we see the optimum for pepsin. We just follow it down. It looks like it's about pH 3 on that graph to me. And if we follow down our trypsin, then that looks like it's about pH 7.8-ish, I would guess. If you're asked to read it on a graph in your A level, you will actually have a nice scale to read. So you won't be estimating like I've just done by eye. But because we've got two enzymes designed to work in different locations in the body, pepsin in an acidic environment, its optimum is on the acid side, whereas trypsin works in just over neutral, okay? As always, I suggest you subscribe to the channel so you can see when more videos are uploaded, and of course head on over to the A-Level Biology website where you can find a range of other resources designed to help you in your A-Level Biology studies.